Good morning to one and all. Today we are going to discuss about the one of the new lesson that is called it as plant tissue culture. Here what is plant tissue culture? Simply we study about tissue culture. Tissue means a group of cells. That group of cells that can be able to cultured in a one of the artificial condition that is called it as a plant tissue culture. That plant tissue culture is one of the technique that can be able to used nowadays for our day-to-day life activity or our survival. Here we have to study elaborative manner in plant tissue culture. What is plant tissue culture? The plant tissue culture is nothing but growing plant protoplast, cell, tissues or organs away from their natural or normal environment. What is the meaning of natural or normal environment? Natural or normal environment means in our field condition. But in tissue culture, we have to use the laboratory condition. That is called it as artificial condition. So this process is called it as, that entire process is called it as tissue culture. Here the tissue culture is otherwise called it as, or it is otherwise known as in vitro. In vitro is nothing but laboratory, lab condition. In an artificial laboratory condition, that is called it as in vitro. The in vitro means a simple meaning, that is nothing but or in glass or in a test tube. In vitro is nothing but a laboratory condition or in a glass or in a test tube. That is a simple meaning for in vitro. Why we have to use the in vitro condition means, in vitro condition only the growing of the plant that can be able to multiplied with the help of tissue culture. Here we have to use a single explant. What is the meaning of explant? The explant is nothing but any part of the plant, whether it is a root, whether it is a stem, whether it is a leaf or flower or fruit, anything, that is called it as explant, that can be able to multiplied with the help of the tissue culture. We have to produce here several thousand plants in a particular period, especially in a very short period. That is called it as a tissue culture. Especially they need some of the controlled condition. That controlled condition is nothing but a temperature, light, cultural medium, nutrient, etc. They can be comes under the controlled condition. This is nothing but plant tissue culture. The plant tissue culture that can be able to serve as a indispensable tool for the regeneration of transgenic plants. Indispensable means there is a basic tool for the formation of transgenic plant. Here in a plant tissue culture, the main application is, most of the applications are there, but in our day-to-day life activity, in our human survival, there is a main applications are there. The first one is clonal propagation. What is the meaning of clonal propagation? Clonal is nothing but as such copy or a duplicate copy of the plant parts. That is nothing but a vegetative propagation. Some of the plants that can be easily cloned. Cloning means the duplicate copy for the parent. That is called it as clonal propagation. Next one is conservation of endangered plant. What is the meaning of endangered plants? The plants that can be extinct, nearly extinct for their life. That is called it as endangered plant, that can be easily conserved. For example, one of the god tree, Vilvam, it is also comes under endangered, but nowadays that can be available in our nursery. Then only conservation of endangered plants. Next one is production of virus free plants. That production of virus free plant is nothing but, for example, virus, they cannot be multiplied in the meristem, especially tip of the plant or tip of the shoot. That is, that virus cannot survive in the tip portion. They can be free from virus. With the help of the meristem culture, we want to produce the virus free plants. For example, banana plant, because a banana plant that can be able to affect by the one of the disease, that is called it as viral disease, that is called it as bunch top of banana. Then it can be able to remedy with the help of production of virus free plants. Next one is germ plasm conservation. The germ plasm conservation is nothing but, the germ is nothing but a gene, the gene that can be able to preserved with the help of plant tissue culture technique, that is called it as germ plasm conservation. Next one is call it as industrial production of secondary metabolites. With the help of the secondary metabolites, we want to produce the number of product, for example, alkaloid, flavonoids, phenols, some of the recombinant proteins. These are all comes under secondary metabolites that can be able to produce with the help of the tissue culture. Then here one of the scientists, Gottlieb Haberland, he was a German botanist. He proposed the concept of totipotency. The totipotency is nothing but, or simply we say, total potentiality of the plant that can be able to produce. That concept was proposed by Gottlieb Haberland. He was the German botanist. He was also a father of plant tissue culture because he proposed the concept of totipotency with the help of mesophyll cells of Lamium purpureum in a culture medium. That culture medium that can be able to helpful for the tissue culture technique. Then only he was called it as a father of tissue culture.